Do you know what is the function of hormones? Do they work as wireless connection as Wi-Fi, or they work as wired connection like broadband? Let's see what are hormones. These are group of small molecules produced at one place and acts on other place. They travel through bloodstream in humans or they act on neighboring cells. But what is the difference between plant hormones and animal hormones? Animal hormones may be synthesized in multiple areas and travel through bloodstream, but plant hormones are not like this. They are synthesized in a single tissue. They act like animal hormones. They work on locally distant site. Okay. What are the functions of these plant hormones? Let us say cell division, cell elongation, differentiation, seed germination, fruit ripening. Ripening means falling. Okay. Stress responses and senescence. Okay. These are the functions of plant hormones. These functions are done by different hormones, not with single hormone. Okay. Let's see how plant hormones are produced and how they are utilized. What are the steps involved in plant hormone production and utilization? The first thing that should be happen, that is biosynthesis. That means production. The second step is storing them in inactive form. So storage. The third step is breakdown. Fourth step is transport. And the final one is signal transduction. Now let's go to the first slide of today's topic. Introduction to plant hormones. I already told you everything about this. They are small organic molecules produced in plant that regulate the growth. Okay. These are in very low concentrations. They act in locally or distant sites. They regulate the cell division, elongation, differentiation. They often interact with other hormones and function combinedly. So we call it as crosstalk, like auxins plus cytokinins. So both are works in growth and cell division. They may act as synergistically or antagonistically. So they work like friends or like enemies. Okay. Major classes of plant hormones. We generally have studied about these hormones. They are auxins, gibberellins, cytokinins, ethylene, abscisic acid. The other growth regulators are brasinosteroids, jasmonic acid, salicylic acid, stingolactones. The second point is general features of hormone regulation. I told you this also. Plant hormone regulation involves 5 main steps. They are biosynthesis, formation of hormone, storage, inactive forms are stored. The third one is transport, movement within the plant. The fourth one, plant perception and transduction. The fifth one is catabolism. That means hormone inactivation. Let's see now about auxin. Auxin biosynthesis. The main natural auxin, we call it as indole 3 acetic acid, that is IAA. It is synthesized in shoot apical meristem. That means it produced at the top. Okay. Also in young leaves, also in developing seeds. So it is produced in 3 areas. Okay. Now let's see how the oxygen is produced. It is mainly synthesized from the tryptophan. We know tryptophan is an amino acid. So auxin is synthesized from the tryptophan. Major pathway, that is the first precursor molecule is tryptophan. The enzyme utilized here is TAA1 enzyme. That means tryptophan amino transferase enzyme. Okay. So it formed indole 3 pyruvic acid. Now the enzyme utilized here is yucca-flavin monoxygenase that formed indole acetic acid. There are other pathways also. This is one major pathway. The other pathways are tryptophan pathway, indole 3 acetamide pathway, tryptophan independent pathway. These are important keywords, so please remember these all. It is stored as inactive conjugates. So any enzyme that stores as inactive form. Okay. The inactive forms are IAA glucose, IAA aspartate, IAA glutamate. The storage occurs in vacuoles, cytoplasm. So this conjugation is helpful in rapid release when required. Okay. If the hormone is required to the plant, then it will be rapidly released if it is in conjugation form. If this hormone is no longer helpful, then it should be inactivated. So let us see how it happens. We call it as breakdown. That means catabolism. We know what is anabolism, catabolism, right? Anabolism means synthesizing, catabolism means utilizing or breakdown. It is inactivated by oxidation process. Auxin, that means indole acetic acid, that is converted to oxindole 3 acetic acid. The enzyme working here is IAA oxidase. There is also second method, auxin that combines with amino acids and it became inactive form. Okay. There is also third step that includes photodegradation. Light can destroy the auxin. Auxin transport is polar and directional. Polar means one end. Directional means from one direction to another direction. It occurs from shoot apex to base. That means from the top to bottom. Okay. These proteins are helpful in transport of auxin. They are pin proteins, auxin efflux carriers. Also, AUX1 lax proteins, influx carriers. So memorize these keywords. Okay. Transport occurs through mainly in parenchyma cells and in the phloem. It is called as non-polar transport. Now let's see physiological effects. Here we can see that how auxin work, how it elongates the cells. It creates acidity in the cells. That's why cell elongates. Auxin activates the proton ATPase. Now cell wall gets acidity. So cell wall expands here. Now cell elongation happens. So what is happening here? First acid is synthesizing. That means protons are increasing. Due to that acid, cell wall size is increasing. So cell elongation is happening. These are the functions of auxin. The first one is apical dominance. It promotes the growth of tip of the plant. So it suppresses the growth of lateral buds. So only promotes the growth of top area. Okay. The second function is phototropism. We know sunflower plants. What they will do? They bend towards the sunlight, right? So this auxin functions like that. It is sun. Please believe it is a sun. Okay. So auxin is present opposite to the sunlight. That causes bending in the bud. By this action, photosynthesis is increases. Okay. The third function of auxin is root inhibition. What are advantageous roots? Do you know? Please believe these are roots. Okay. Generally, where will be the roots? They are at bottom, right? Advantageous roots forms from the leaves or stem or nodes. Okay. They are different from the normal roots. They are not producing in the bottom of the root, right? They can produce from anywhere. So we call it as advantageous roots. So auxins are promoting these roots. Vascular differentiation. So it stimulates the differentiation of xylem and phloem. Their structure may be different or maybe some tissue may be between the both tissues, right? Mechanism of action. Auxin signaling uses SCF ubiquitin pathway. These are keywords. Please remember this. I cannot explain every word. Okay. The first step is auxin bind to TIR1 receptor. So now auxin TIR1 complex is formed here, and again this complex bind with auxin proteins. Aux or indolestic acid proteins are degraded via. Why it is degraded by proteasome? What is happening here? Did you understand anything? When auxin is stored in inactive form and plant needs auxin, so it should react with proteasome. It should be degraded, and now auxin responsive genes are expressed. Now auxin releases into the plant. So why it is happening? To produce the auxin. It happens rapidly, not slow process. This proteasome is of 26S proteasome, right? When auxin present heavily, SCF ubiquitin inactivated and this processor become reversed. So there is no degradation, there is no genes expression, no auxin production, right? Remember these are keywords. I cannot explain you everything, but I am explaining. You have to mug up this. You have to memorize this all. Nobody can help you with this. This total plant physiology is like this only. We have to memorize so many things. Here explanation is very less and memorization is very high.